Uh, Warm regards to all the organizing committee of this conference. I'm very thankful to you for providing me an opportunity to present and research article on evaluation of carcinogenic nitrosamine impurity labels in olmesartan tablet using a sensitive LC-MS method. Let me introduce myself. My name is Namdev Shamrao Pawar, and I have conducted this study under the guidance of Dr. Aparna Bharadwaj. I am a PhD scholar. So let us understand what N-nitroso impurities or nitrosamines. These are basically a small compound with molecular weight of around 75 and 120. And these are mutagenic human carcinogens. And these are very fatal and can cause cancer in the patient who are in a prolonged use. There are so many nitrosamines present, out of which N-nitrosodimethylamine and N-nitrosodiethylamine, that is NDMA and NDEA, were practically found in some ARBs or certain groups, certain medications. So nitroso compounds are very well known compound. Uh, they were found in they are basically found in water, food, tobacco, pesticides, or plastic, or we can say cosmetics. But in July 2018, it was discovered in pharmaceuticals. So uh, on the basis of this discovery, all the regulatories like ICH, EMA, or USFDA, they have highly concerns about this because it is dealing with the human safety, uh, and it was then they decided the specification limit for that, and they given the guidance for that. So this traceable amount of nitrosamine impurity is responsible for alteration of safety and efficacy, and these compounds are known as cohort of concern given by WHO IARC committee. As I said earlier, ARB medications like candesartan, olmesartan, irbesartan, they were recalled from the market due to the presence of this carcinogenic impurity. So there is a high need to estimate them in drug products or drug substances. From drug product, it should be properly estimated since they are very trace compounds, and for proper estimation, we required a simple, selective, reliable, reproducible method to fulfill all the standards. So here I set my aim to uh, develop and validate a method which can identify and quantify the trace amount of this NDMA and NDEA from one of the sartans, that is olmesartan. Uh, to move further, I have developed a analytical method, then I validated it, and then I checked the validated method with several marketed drugs. So before starting, I have uh, I have initiated my method development with tuning up the LCMS system. I have uh, uh, set the compound parameters and source parameters, and then on the basis of that, the estimation was carried out. And with several trials, with different trials, I have selected a mobile phase and column that was best suited for this method or estimation of this stress level impurity. Then I have uh, extracted the olmesartan, NDMA, NDEA from drug product, that is tablet doses form, using various solvent, and finally I have extracted it very well from methanol by using methanol. Sorry. Then uh, once I set the method or developed the method, I have validated it properly. for further analysis, and the validation parameters are taken from the guidance of ICH, that is Q2R2. Here we can see the peak and proper resolution of NDMA and NDEA. Here is the retention of NDMA and here is the retention time of NDEA. Then I have decided the LOD and LOQ for that and run the specificity using a blank compound, 100% solution and a 100% solution in tablet. So the results from the tablet blank is in front of you. Here we can see there is no interference from the coeluents or other excipients, binders, or any coating at the retention time of NDMA and NDA. So the method is specific. Then I move forward further, and I have decided a range for linearity of 5 different points, and 6th point is LOQ. These 5 different points are at the specification level, 100% level, then 50% level, 120% level, and within that 80 and 120% level, and LOQ. So here in linearity, we can see that the correlation coefficient is more than 0.99. This is acceptable. Now I move further to find out the recovery, and recovery is calculated on the basis of accuracy. Here I prepared a solutions of 50%, 100%, and 150% in triplicates, and find overall recovery 89.22 and 88.34 for NDMA and NDEA respectively. And this is very well accepted since the acceptance criteria is 70 to 130%. Then I move to the precision of that method, reproducibility of the method. I have conducted method precision and intermediate precision with 12 replicates and 18 replicates respectively. For method precision and intermediate precision, the 6 replicates were run on one day, and for method precision, 6 replicates on another day. For intermediate precision, 6 replicates on one day and 12 replicates on another day. Here are the results of limit of detection and limit of quantification. The signal to noise ratio, it is calculated on the basis of signal to noise ratio. Signal to noise ratio should be more than 3 at LOD level and should be more than 10 at LOQ level. And at LOQ level, it was the suitability of sensitivity of that limit of quantification level was checked with the 6 replicate run uh, uh, for NDMA and NDEA. Here I found that the method is very well suited and can identify and quantify the trace level of nitroso group, this NDMA and NDEA. I have applied it to several marketed drugs, and I found that there was no interference from any other compound or any impurities, any traces of impurities uh, in all the drugs, all the tablets. Here are some bibliography which I search under literature search. So here I conclude my presentation, and I say that my method is very well suited for the estimation of NDMA and NDEA from olmesartan tablet. Uh, Thank you so much for uh, giving me a opportunity to present this, and all the best for the conference. Thank you very much, sir.